The T helper cells are a type of T cell that play an important role in the immune system, particularly in the adaptive immune system. They help the activity of other immune cells by releasing T cell cytokines. These cells help suppress or regulate immune responses. They are essential in B-cell antibody class switching, in the activation and growth of cytotoxic T cells, and in maximizing bactericidal activity of phagocytes such as macrophages. Mature Th cells express the surface protein CD4 and are referred to as CD4 plus T cells. CD4 plus T cells are generally treated as having a predefined role as helper T cells within the immune system. For example, when an antigen-presenting cell expresses an antigen on MHC class II, a CD4 plus cell will aid those cells through a combination of cell-to-cell interactions and through cytokines. Nevertheless, there are rare exceptions. For example, subgroups of regulatory T cells, natural killer cells, and cytotoxic T cells express CD4. All of the latter CD4 plus T cell groups are not considered T helper cells. The importance of helper T cells can be seen from HIV, a virus that infects CD4 plus cells. Towards the end of an HIV infection, the number of functional CD4 plus T cells falls, which leads to the symptomatic stage of infection known as the acquired immunodeficiency syndrome. There are also some rare disorders that result in the absence or dysfunction of CD4 plus T cells. These disorders produce similar symptoms, and many of these are fatal. Activation of Norvay helper T cells. Following T cell development, matured Norvay T cells leave the thymus and begin to spread throughout the body, including the lymph nodes. Like all T cells, they express the T cell receptor CD3 complex. The T cell receptor consists of both constant and variable regions. The variable region determines what antigen the T cell can respond to. CD4 plus T cells have TCRs with an affinity for class II MHC, and CD4 is involved in determining MHC affinity during maturation in the thymus. Class II MHC proteins are generally only found on the surface of specialized antigen-presenting cells. Specialized antigen-presenting cells are primarily dendritic cells, macrophages and B cells, although B cells are the only cell group that expresses MHC class II constitutively. Some APCs also bind native antigens to their surface, such as follicular dendritic cells, but unprocessed antigens do not interact with T cells and are not involved in their activation. The antigens that bind to MHC proteins are always short peptides, 8 to 10 amino acids long for MHC class I, and up to 25 or so for MHC class II. Recognition: during an immune response, professional antigen-presenting cells endocyte as foreign material, which undergoes processing, then travel from the infection site to the lymph nodes. Once at the lymph nodes, the APC begin to present antigen peptides that are bound to class II MHC, allowing CD4 plus T cells that express the specific TCRs against the peptide MHC complex to activate. When a Th cell encounters and recognizes the antigen on an APC, the TCR CD3 complex binds strongly to the peptide MHC complex present on the surface of professional APCs. CD4, a co-receptor of the TCR complex, also binds to a different section of the MHC molecule. These interactions bring these proteins closer together, allowing the intracellular kinases present on the TCR, CD3 and CD4 proteins to activate each other via phosphorylation. With the assistance of a phosphatase present on the intracellular section of CD45, these molecules activate major Th cell intracellular pathways. These active pathways are known as signal 1 of T cell activation, as it is the first and primary pro-activation signal in a Th cell. Upon subsequent encounters with a given antigen, memory T cells are reactivated using the same TCR pathways. The binding of the antigen MHC to the TCR complex and CD4 may also help the APC and the Th cell adhere during Th cell activation, but the integrin protein LFA1 on the T cell and ICAM on the APC are the primary molecules of adhesion in this cell interaction. It is unknown what role the relatively bulky extracellular region of CD45 plays during cell interactions, but CD45 has various isoforms that change in size depending on the Th cell's activation and maturation status. For example, CD45 shortens in length following Th activation, but whether this change in length influences activation is unknown. It has been proposed that the larger CD45 or A+ may decrease the accessibility of the T-cell receptor for the antigen MHC molecule, thereby necessitating an increase in the affinity of the T-cell for activation. Once the activation has occurred, however, CD45 shortens, allowing easier interactions and activation as an effector T helper cell. Verification: having received the first TCR-CD3 signal, the Norvay T cell must activate a second independent biochemical pathway, known as signal 2. This verification step is a protective measure to ensure that a T cell is responding to a foreign antigen. If this second signal is not present during initial antigen exposure, the T cell presumes that it is autoreactive. This results in the cell becoming anergic. Anergic cells will not respond to any antigen in the future, even if both signals are present later on. These cells are generally believed to circulate throughout the body with no value until they undergo apoptosis. The second signal involves an interaction between CD28 on the CD4 plus T cell and the protein CD80 or CD86 on the professional APCs. Both CD80 and CD86 activate the CD28 receptor. These proteins are also known as co-stimulatory molecules. Although the verification stage is necessary for the activation of Norvay helper T cells, the importance of this stage is best demonstrated during the similar activation mechanism of CD8 plus cytotoxic T cells. As Norvay CD8 plus T cells have no true bias towards foreign sources, these T cells must rely on the activation of CD28 for confirmation that they recognize a foreign antigen. CD28 plays an important role in decreasing the risk of T cell autoimmunity against host antigens. Once the Norvay T cell has both pathways activated, the biochemical changes induced by signal 1 are altered, allowing the cell to activate instead of energize. The second signal is then obsolete. Only the first signal is necessary for future activation. This is also true for memory T cells, which is one example of learned immunity. Faster responses occur upon reinfection because memory T cells have already undergone confirmation and can produce effector cells much sooner. Proliferation: once the two-signal activation is complete, the T helper cell then allows itself to proliferate. It achieves this by releasing a potent T-cell growth factor called interleukin-2, which acts upon itself in an autocrine fashion. Activated T-cells also produce the alpha subunit of the IL-2 receptor, enabling a fully functional receptor that can bind with IL-2, which in turn activates the T-cell's proliferation pathways. The autocrine or paracrine secretion of IL-2 combine that same Th cell or neighboring Ths via the IL-2 are thus driving proliferation and clonal expansion. The Th cells receiving both signals of activation will then become Tho cells that secrete IL-2, IL-4 and interferon gamma. The Tho cells will then differentiate into Th1 or Th2 cells depending on cytokine environment. IFNI³ drives Th1 cell production while IL-10 and IL-4 inhibit Th1 cell production. Conversely, IL-4 drives Th2 cell production and IFNI³ inhibits Th2 cells. It should be noted that these cytokines are pleiotropic and carry out many other functions of the immune response. Maturation: after many cell generations, the Th cells' progenitors differentiate into effector Th cells, memory Th cells, and regulatory Th cells. Effector Th cells secrete cytokines, proteins or peptides that stimulate or interact with other leukocytes, including Th cells. Memory Th cells retain the antigen affinity of the originally activated T cell, and are used to act as later effector cells during a second immune response. Regulatory T cells do not promote immune function, but act to decrease it instead. Despite their low numbers during an infection, These cells are believed to play an important role in the self-limitation of the immune system. They have been shown to prevent the development of various autoimmune diseases. Determination of the effector T-cell response. Helper T-cells are capable of influencing a variety of immune cells, and the T-cell response generated can be essential for a successful outcome from infection. In order to be effective, helper T-cells must determine which cytokines will allow the immune system to be most useful or beneficial for the host. Understanding exactly how helper T-cells respond to immune challenges is currently of major interest in immunology, because such knowledge may be very useful in the treatment of disease and in increasing the effectiveness of vaccination. Th1/Th2 model for helper T-cells. Proliferating helper T-cells that develop into effector T-cells differentiate into two major subtypes of cells known as Th1 and Th2 cells. Th1 helper cells are the host immunity effectors against intracellular bacteria and protozoa. They are triggered by IL-12, IL-2 and their effector cytokine is IFNI-cubed. The main effector cells of Th1 immunity are macrophages as well as CD8 T cells, IgG cells, and IFNI-cubed CD4 T cells. The key Th1 transcription factors are STAT4 and Tbet. IFNI-cubed secreted by CD4 T cells can activate macrophages to phagocytose and digest intracellular bacteria and protozoa. In addition, IFNI-cubed can activate iNOS to produce NOx free radicals to directly kill intracellular bacteria and protozoa. Th1 overactivation against autoantigens will cause type 4 delayed type hypersensitivity. Tuberculin reaction or type 1 diabetes belong to this category of autoimmunity. Th2 helper cell are the host immunity effectors against extracellular parasites including helminths. They are triggered by IL-4 and their effector cytokines are IL-4, IL-5, IL-9, IL-10 and IL-13. The main effector cells are eosinophils, basophils, and mast cells as well as B cells, and IL-4/IL-5 CD4 T cells. The key Th2 transcription factors are STAT6 and GATAs. IL-4 is the positive feedback cytokine for Th2 cells differentiation. Besides, IL-4 stimulates B cells to produce IgE antibodies, which in turn stimulate mast cells to release histamine, serotonin, and leukotriene to cause bronchoconstriction, intestinal peristalsis, gastric fluid acidification to expel helminths. IL-5 from CD4 T cells will activate eosinophils to attack helminths. IL-10 suppresses Th1 cells differentiation and function of dendritic cells. Th2 overactivation against autoantigen will cause type 1 IgE-mediated allergy and hypersensitivity. Allergic rhinitis, atopic dermatitis, and asthma belong to this category of autoimmunity. While we know about the types of cytokine patterns helper T cells tend to produce, we understand less about how the patterns themselves are decided. Various evidence suggests that the type of APC presenting the antigen to the T cell has a major influence on its profile. Other evidence suggests that the concentration of antigen presented to the T cell during primary activation influences its choice. The presence of some cytokines will also influence the response that will eventually be generated, but our understanding is nowhere near complete. Th17 helper cells. Th17 helper cells mediate host immunity against extracellular bacteria and fungi. This effector cell subtype is triggered by IL-6 and TGF-beta or IL-23 and IL-1-beta. Its main effector cytokines are IL-17A, IL-21, and IL-22. The main Th17 effector cells are neutrophils as well as IgM-IgA-B cells, and IL-17 CD4 T cells. The key Th17 transcription factors are STAT3 and ROG. The Th17 cells can alter their differentiation program, ultimately giving rise to either protective or pro-inflammatory pathogenic cells. The protective and non-pathogenic Th17 cells induced by IL-6 and TGF-beta are termed as TREG17 cells. The pathogenic Th17 cells are induced by IL-23 and IL-1 beta. TNF-alpha can activate neutrophils to kill extracellular bacteria and fungi. Besides, IL-6 can upregulate the complement system to directly kill extracellular bacteria and fungi. Th17 overactivation against autoantigen will cause type 3 immune complex and complement-mediated hypersensitivity. Rheumatoid arthritis or Arthus reaction belong to this category. Bone erosion caused by mature osteoclast cells is common in patients with rheumatoid arthritis. Activated T helper cells such as Th1, Th2, and Th17 are found in the synovial cavity during the time of inflammation due to rheumatoid arthritis. The known mechanisms associated with the differentiation of osteoclast precursors into mature osteoclasts involve the signaling molecules produced by immune associate cells, as well as the direct cell-to-cell contact of osteoblasts and osteoclast precursors. However, it has been suggested that Th17 can also play a more major role in osteoclast differentiation via cell-to-cell contact with osteoclast precursors. Thi plus or minus I squared helper cells. Thi plus or minus I squared helper cells provide the host immunity against viruses. Their differentiation is triggered by IFN-alpha-beta or IL-10. Their key effector cytokine is IL-10. Their main effector cells are NK cells as well as CD8 T cells, IgG cells, and IL-10 CD4 T cells. The key Thi plus or minus I squared transcription factors are STAT1 and STAT3 as well as IRFs. IL-10 from CD4 T cells activate NK cells ADCC to apoptose virus-infected cells and to induce host as well as viral DNA fragmentation. IFN-alpha-beta can suppress transcription to avoid virus replication and transmission. Overactivation of Thi plus or minus I squared against autoantigen will cause type 2 antibody-dependent cytotoxic hypersensitivity. Myasthenia gravis or Graves' disease belong to this category. Limitations to the Th1/Th2 model. The interactions between cytokines from the Th1/Th2 model can be more complicated in some animals. For example, the Th2 cytokine IL-10 inhibits cytokine production of both Th subsets in humans. Human IL-10 suppresses the proliferation and cytokine production of all T cells and the activity of macrophages, but continues to stimulate plasma cells, ensuring that antibody production still occurs. As such, HEL10 is not believed to truly promote the Th2 response in humans, but acts to prevent overstimulation of helper T cells while still maximizing the production of antibodies. There are also other types of T cells that can influence the expression and activation of helper T cells, such as natural regulatory T cells, along with less common cytokine profiles such as the Th3 subset of helper T cells. Terms such as regulatory and suppression have become ambiguous after the discovery that helper CD4 plus T cells are also capable of regulating their own responses outside of dedicated regulatory T cells. One major difference between regulatory T cells and effector T cells is that regulatory T cells typically serve to modulate and deactivate the immune response, while effector T cell groups usually begin with immune-promoting cytokines and then switch to inhibitory cytokines later in their life cycle. The latter is a feature of Th3 cells, which transform into a regulatory subset after its initial activation and cytokine production. Both regulatory T cells and Th3 cells produce the cytokine transforming growth factor beta and IL-10. Both cytokines are inhibitory to helper T cells. TGFI² suppresses the activity of most of the immune system. There is evidence to suggest that TGFI² may not suppress activated Th2 cells as effectively as it might suppress naive cells, but it is not typically considered a Th2 cytokine. The characterization of another novel T helper subtype, T helper 17 cells, has cast further doubt on the basic Th1/Th2 model. These IL-17 producing cells were initially described as a pathogenic population implicated in autoimmunity but are now thought to have their own distinct effector and regulatory functions. Of note, Recent evidence suggests that functional plasticity is an intrinsic capacity of T helper cells. Indeed, a study in mice demonstrated that Th17 cells transform into Th1 cells in vivo. A subsequent study furthermore showed that extensive T helper cell plasticity is also prominent in man. Many of the cytokines in this article are also expressed by other immune cells, and it is becoming clear that while the original Th1/Th2 model is enlightening and gives insight into the functions of helper T cells, it is far too simple to define its entire role or actions. Some immunologists question the model completely, as some in vivo studies suggest that individual helper T cells usually do not match the specific cytokine profiles of the Th model, and many cells express cytokines from both profiles. That said, the Th model has still played an important part in developing our understanding of the roles and behavior of helper T cells and the cytokines they produce during an immune response. Recent scientific studies by Stockinger et al. Revealed that another T helper subset may exist. Th9 cells are claimed to be an IL-9 producing T cell subset focused on defending helminth infections. Role of helper T cells in disease. Considering the diverse and important role helper T cells play in the immune system, it is not surprising that these cells often influence the immune response against disease. They also appear to make occasional mistakes, or generate responses that would be politely considered non-beneficial. In the worst-case scenario, the helper T cell response could lead to a disaster and the fatality of the host. Fortunately, this is a very rare occurrence. Helper T cells and hypersensitivity. The immune system must achieve a balance of sensitivity in order to respond to foreign antigens without responding to the antigens of the host itself. When the immune system responds to very low levels of antigen that it usually shouldn't respond to, a hypersensitivity response occurs. Hypersensitivity is believed to be the cause of allergy and some autoimmune disease. Hypersensitivity reactions can be divided into 4 types. Type 1 hypersensitivity includes common immune disorders such as asthma, allergic rhinitis, eczema, urticaria and anaphylaxis. These reactions all involve IgE antibodies, which require a Th2 response during helper T cell development. Preventive treatments, such as corticosteroids and montelukast, focus on suppressing mast cells or other allergic cells. T cells do not play a primary role during the actual inflammatory response. It's important to note that the numeral allocation of hypersensitivity types does not correlate to the response in the Th model. Type 2 and Type 3 hypersensitivity both involve complications from autoimmune or low-affinity antibodies. In both of these reactions, T cells may play an accomplice role in generating these autospecific antibodies, although some of these reactions under Type 2 hypersensitivity would be considered normal in a healthy immune system. The understanding of the role of helper T cells in these responses is limited, but it is generally thought that Th2 cytokines would promote such disorders. For example, studies have suggested that lupus and other autoimmune diseases of similar nature can be linked to the production of Th2 cytokines. Type 4 hypersensitivity, also known as delayed type hypersensitivity, are caused via the overstimulation of immune cells, commonly lymphocytes and macrophages, resulting in chronic inflammation and cytokine release. Antibodies do not play a direct role in this allergy type. T cells play an important role in this hypersensitivity, as they activate against the stimulus itself and promote the activation of other cells, particularly macrophages via Th1 cytokines. Other cellular hypersensitivities include cytotoxic T-cell-mediated autoimmune disease, and a similar phenomenon. Transplant rejection. Helper T cells are required to fuel the development of these diseases. In order to create sufficient autoreactive killer T cells, interleukin-2 must be produced, and this is supplied by CD4 plus T cells. CD4 plus T cells can also stimulate cells such as natural killer cells and macrophages via cytokines such as interferon gamma, encouraging these cytotoxic cells to kill host cells in certain circumstances. The mechanism that killer T cells use during autoimmunity is almost identical to their response against viruses, and some viruses have been accused of causing autoimmune diseases such as type 1 diabetes mellitus. Cellular autoimmune disease occurs because the host antigen recognition systems fail, and the immune system believes, by mistake, that a host antigen is foreign. As a result, the CD8+ T cells treat the host cell presenting that antigen as infected, and go on to destroy all host cells that express that antigen. Some of this section is a simplification. Many autoimmune diseases are more complex. A well-known example is rheumatoid arthritis, where both antibodies and immune cells are known to play a role in the pathology. Generally, the immunology of most autoimmune diseases is not well understood. HIV infection, perhaps the best example of the importance of CD4+ T cells, is demonstrated with human immunodeficiency virus infection. HIV targets cells that express CD4, and can infect macrophages, dendritic cells and CD4+ T cells. It has been proposed that during the non-symptomatic phase of HIV infection, the virus has a relatively low affinity towards T cells, resulting in a slow kill rate of CD4+ T cells by the immune system. This is initially compensated for via the production of new helper T cells from the thymus. Once the virus becomes lymphotropic, however, it begins to infect CD4+ T cells far more efficiently, and the immune system is overwhelmed. At this point, functional CD4+ T cell levels begin to decrease, eventually to a point where the CD4+ T cell population is too small to recognize the full range of antigens that could potentially be detected. The lack of full antigen cover results in the core symptoms of acquired immune deficiency syndrome. CD40 cell depletion during AIDS allows various pathogens to escape T cell recognition, thus allowing opportunistic infections that would normally elicit a helper T cell response to bypass the immune system. While these complete bypass situations only occur when the helper T cell response is absolutely necessary for infection clearance, most infections increase in severity and/or duration because the immune system's helper T cells provide a weaker contribution to a less efficient immune response. Two components of the immune system are particularly affected in AIDS, due to its CD4+ T cell dependency. CD8+ T cells are not stimulated as effectively during the AIDS stage of HIV infection, making AIDS patients very susceptible to most viruses, including HIV itself. This decline in killing of CD4+ T cells results in the virus being produced for a longer period, increasing the proliferation of the virus, and accelerating the development of the disease. Antibody class switching declines significantly once helper T cell function fails. The immune system loses its ability to improve the affinity of their antibodies, and are unable to generate B cells that can produce antibody groups such as IgG and IgA. These effects are primarily due to the loss of any helper T cell that can interact with the B lymphocyte correctly. Another symptom of AIDS is the reduction in antibody levels due to a decrease in Th2 cytokines. All of these complications result in an increased susceptibility to aggressive bacterial infections, especially in areas of the body not accessible by IgM antibodies. If the patient does not respond to HIV treatment, they will succumb usually to either cancers or infections. The immune system finally reaches a point where it is no longer coordinated or stimulated enough to deal with the disease. Inhibition of CD40 cell expansion during HIV infection is also due to microbial translocation in an IL-10-dependent way. Indeed, triggering PD-1, expressed on monocytes and upregulated upon monocytes activation, by its ligand PD-L1 induces IL-10 production which inhibits CD40 cell function. See also, CD4+ T cells and anti-tumor immunity, references. Kano Y, Vaid G, Hirahara K, Singleton K and O'Shea J.J. 2012. Transcriptional and epigenetic control of T helper cell specification, molecular mechanisms underlying commitment and plasticity. Annual Review of Immunology 30, 707-731. In SLE patients, T helper type 1 polarization is more abundant rather than type 2, where the main cytokine profile are gamma from T cells and its consequent interferon alpha high-range production by DC. External links: T cell group, Cardiff University.